students who conducted their PhD research over the last couple of years and are either done or very close to being actually defending their PhD. This lectureship was established through funds donated by Samuel Eiduson, and in honor of Samuel Eiduson, who was the first chair of the NSIDP, the Graduate Neuroscience Interdepartmental Program here at UCLA. He chaired it from 1973 until 1985. And in his honor, he established this lectureship, which is given to particularly remarkable research over the course of their PhD. And as I said, in honor of the excellence of our graduate students, the committee just couldn't decide this year who was more worthwhile, and maybe everybody who's on the call or in here can sort of see what they think. Personally, I think having seen their research and having seen their presentations, I think they're really, both of them highly selected, highly qualified candidates. So the first lecture, the first part will be given by Peter Schuette, who worked in the lab of Adhikari. I always stumble over your last name, I'm very sorry, and Jonathan Cow. And the second one will be given by Anna Sias, who conducted her research with Kate Wassum. And I congratulate both of you, Peter and Anna, and in honor of your lecture, your selection, I present you with, maybe we can stand just in front here. Yeah. Stand within this.
Right,
so
gotcha.
maybe I can just give that to you, to both of you. Would that work? Yes. Okay. And so without further ado, I think we're going to start with Peter, introduced by Adhikari. How I got your name.
Thanks, Felix.
Thank you, Felix, for selecting these amazing students with the committee for this prize. So now it's my great pleasure and honor to introduce my first graduate student, Peter Schuette. And he had a very interesting story. So he started with an undergrad in visual arts. So he was doing paintings and sculptures and conceptual, complicated art that I don't understand. And then he went from that to a musical career. Then he played colorful keyboards like this one. And then after that, he was involved in some kind of pagan religious ritual of some sort. And then he had a reorientation in life. So he was like, I can't stay like this and smoke with my hands up in the air forever. So I need to do something. And then he spent some time finding himself. So you can see he's looking at himself in the TV with his mouth open, very insightful times. And then he fell into some dark, depressive times in which he decided to grow a mustache for some reason. And then he's walked around like this. But I also had my dark times with also mustache. But then he hopefully rose through the occasion and then became a mustache-free neuroscientist. And here we are in a representative photo wearing a lab coat, discussing how DAPI staining is very nice. And then after all his accomplishments, we had lots of neuroscience parties to celebrate his many discoveries. And just to give you a quick run through, so when he joined the lab, when we're applying for fellowships and things, asked, oh, we can list your awards. Do you have any awards? And he went, no, I have no awards, and I'm very sad. And then now at the end of the completion of this PhD, he has so many awards. Like he won the National Science Foundation Award, the UCLA Affiliates Award, the UCLA Excellence in Life Sciences Research, which is not a neuroscience awards, but it's competing with all the life sciences students. So he beat people that study salmon migration and botany and all sorts of strange things. And then now today he won the Eiduson Award, which is the lecture you guys will see now. And he also still kept up with his artistic background and won the first place in science art from the UCLA library last year. And even more impressively, he also won second place in science art from the UCLA library in the same year. So that's really very impressive. And he didn't just win awards, but he also published very well and is extremely productive. So he has first author papers in Neuron, eLife, and Scientific Reports and Journal of Neuroscience. And he's also a co-author because he collaborated with everyone because he's very generous with his time and resources. So he got co-authorship papers in eLife and Journal of Neuroscience, and he'll be a future co-author in some papers that are currently submitted, such as papers in eLife and Nature Communications. So today, he will talk about mostly his first author papers in Neuron and eLife. And as you can see, even half of the stuff that he didn't present today would be almost enough to graduate a whole different person. So Peter, just dazzle them with your amazing work. And this is not one of those occasions, sorry, where you are kind of going to the bottom of the barrel to figure out what to say in a lecture, but it's more like a embarrassment of riches where there's so much to choose from, and it's like these pirate chests that always have the treasure overflowing from them. And so you're lucky to see Peter's cool work today. And now there's another thing. One time when he was presenting his data, his data was so hot that literally the fire alarm went off in his building, and then people had to be evacuated and everything. It's a real thing that happened during a committee meeting that we had over Zoom. So now you'll see the hot data from Peter.
Thanks, Avi. It's a real honor to be presenting for this award, and just carrying the torch for people that had mustaches at one point and decided to shave them. All it takes is really just the will and a razor, and you can change your life. So just, for anybody out there, it's possible. So today I'll be presenting on this topic, the coordination of escape and spatial navigation circuits and how they orchestrate versatile flight from multimodal threats. The first chunk of data here was published in Neuron last year in this article. So as you all probably are aware, animals face lethal threats, and this often occurs in geometrically complicated environments. You can see here this cat is trying to catch a rat. The rat is very good at hiding. It seems like it might be the end though. And then lo and behold, performs an amazing escape that you could never predict. So this just to say, escaping from threats can require quite adaptive actions. So for instance, if you're stuck in a house on fire, God forbid, if you're in the kitchen, you would escape out the door. And if you're in the den, you might go out the window. So, escape is adaptive. And we will refer to this throughout the rest of this lecture as context-specific escape where the context is crucial to achieving successful escape. As it turns out, context-specific escape is actually also entertaining, and they make whole movies about it. There's this movie, it's like a little art house film from the mid-90s called Speed, where Keanu Reeves has to escape, everybody has to escape off a bus without it going below 50 miles an hour. So, context-specific escape, it happens, it's entertaining, and it's essential to survival, oftentimes. So in contrast to context-specific escape, sometimes you might need to flee an environment from simpler environments, like an empty box. And this is the way that escape is generally studied in the lab. In these cases, the mouse could run in, say, any direction and achieve safety. So we'll call this simple stereotyped escape. And early on, we hypothesized that this nucleus in the hypothalamus, the dorsal premammillary nucleus, or PMD, is particularly well positioned to modulate both the context-specific and simple stereotype varieties of escape. Here's the PMD in a circuit diagram of the medial hypothalamic defensive circuit. So each of these nodes kind of controls various aspects of defensive behavior. And if you zoom out, here's the PMD in sort of a larger circuit diagram. You can see that it's uniquely connected in ways that would help it achieve this kind of escape behavior I've been talking about. So it interfaces between spatial navigation and defensive circuits. You can see it receives inputs from the hippocampus through the septum, also from the amygdala, where things like the predator odor could be conveyed. And it also has two key outputs to the dorsolateral periaqueductal gray, or the DLPAG, and the anteromedial ventral thalamic nucleus, or AMV. Now, the DLPAG is very strongly implicated with escape. It's been studied for years in regards to the way it controls escape. And activating this region causes flight, inhibiting it impairs threat-induced escape. Now, the AMV, on the other hand, has been shown that when lesioned can impair spatial navigation and contextual memory. and it encodes head direction and projects to navigation regions. So considering all of this connectivity, we hypothesized that the PMD could be orchestrating the coordination that's necessary of contextual navigation and flight for strategic context-specific escape. So before we did any behavioral manipulations, we characterized the cells in this region. We found that the majority expressed cholecystokinin, so they were CCK positive, and they were glutamatergic. So with this in mind, we were able to target cells in the PMD with excellent cell type specificity. As you can see that the PMd-cck cells are quite restricted within the bounds of the hypothalamic nucleus. So using this, we asked our first question, is the PMD necessary and sufficient to control context-specific escape? And this is an assay that we use to get at this question. We place a mouse in a cage with a tethered rat, and the mouse will explore against its own best interests, then runs up the ladder and hides in the burrow. So it would probably stay in the burrow the whole time, but after a minute, we gently coax it down to the floor of the enclosure, and the second trial starts. And here we're counting, the primary metric is the number of escapes. So we're just seeing how many times will the mouse escape in this environment in the course of 20 minutes. So we chose this specific layout because it's very unlikely that the mouse will successfully escape by running randomly in any given direction. It needs to know where the bottom of the ladder is and where the burrow is located to successfully escape and achieve safety up here. So the way that we did this, we habituated the mouse for a few days. We exposed it to a control threat, which in this case was a toy rat, and then the real threat, alternating CNO and saline injections. And as hypothesized, we found that for the inhibitory and excitatory dread cohorts, we respectively decreased and increased the number of escapes. Crucially, we did not see this for the control conditions, the toy rat, indicating that this wasn't just some byproduct of an increase in exploratory drive. In fact, the threat needed to be in the environment to sort of prompt this behavior. So the next obvious question was, is the PMD also necessary and sufficient to control panic-related escape? So to get at this question, we performed a couple of assays in geometrically similar environments. On the left, we exposed the mouse to a heated floor. It was 43 Celsius, uncomfortable enough that the mouse would want to climb up this hose. But it couldn't stay up there forever, so it would turn around and come back down to the floor and then climb again. So here we're counting the number of climbs. And on the right, we exposed the mouse to a panicogenic agent, CO2. It's 15% CO2. And it's not in the video, but there should be a hose here too. This is sort of our glamour video of the assay, but we forgot to put the hose in. But anyway, in this context, with the CO2, it sort of induces the mouse to jump. It performs these escape jumps because the threat is so great. I guess it creates the feeling of asphyxiation. So similar to before, we have two cohorts, the inhibitory and excitatory dread cohorts. We habituated to each environment, exposed them to control threat, which would be a room temperature plate and air rather than CO2, and then the real threat, alternating saline and CNO injections. And similar to before, we actually found that in both cases, we could bidirectionally modulate the number of escapes for each assay. So we checked some other things. These results were not attributable to things like pain perception. We did a latency to pain response test, and there was no effect of the dreads. We performed it wasn't due to anxiety as measured as the fraction of time in the center of the empty open field, and locomotion, the velocity in that open field. So now we went on to perform a few experiments. These are some of my favorites, where we asked, does optogenetic activation of these same cells cause escape across a variety of contexts, which each require their own escape strategy? So it's sort of like how flexible is this, are these behaviors that we're seeing? Will it adapt to any kind of escape route that we present the mouse with? So here we have the first assay. It's the inescapable empty box. So we place a mouse in a box and we stimulate the PMd-cck neurons with channel rhodopsin. The laser's on, and really there's no way to escape the box, as the name implies. So you can see the mouse is just performing some escape jumps, and that's sort of the best it can do to try to get out of the box. However, as soon as we introduce a rope into the enclosure and turn the laser on, the mouse pretty quickly finds the rope and escapes the box. And one thing to note here, this is the first time the mouse has ever been introduced to any of these environments, so it has to solve these problems as it's presented with them. Here we have an upward step assay where there's a couple of steps, and one leads to the exit, one does not. And the laser once the laser turns on, the mouse pretty quickly figures out which stairway to climb to escape the enclosure. And lastly, just to make sure there wasn't some kind of upward specific aspect of this behavior, we did a downward step assay, placing the mouse at the top of the stairs, and it indeed escaped downwards and around the corner. So back to the inescapable empty box, we found that across two different laser intensities, actually, so both 1 and 4 milliwatt laser intensities would induce jumping, and neither would induce freezing. So it seemed very specific to escape behavior rather than freezing or other defensive behaviors. And as soon as an exit route was introduced, all of our CCK mice escaped within 15 or 20 seconds of light on. So this is a robust effect, and it's highly versatile. The mouse could adapt it to whatever environment it was in, and if there was a way to escape, it would escape. So now this is some of these assays have obviously not been tested in other labs. We've sort of made them up. So we are curious if any other regions might control this specific kind of context, this context-specific escape that we're looking at here. To develop a list of some candidate regions, our partner lab, the Cantorus lab, performed some FOSS studies, exposing mice to a cat, a rat, and CO2, and measured the difference in the increase in FOSS from control. And you can see here that there's a number of brain regions that showed an increase, a significant increase in FOSS, and many of them are sort of expected ones, the PMD, the PAG. There's the medial hypothalamic defensive system is included in here, the other two nuclei that I spoke about earlier. So we expressed channel rhodopsin in each of these regions and in wild type mice, and stimulated first in the empty open box. You can see here that a few of these regions, stimulation caused jumping. However, as soon as we tested them in the upward step, you can see that the PMD was the only one that caused successful escape. So while some of the regions might cause escape-related behavior, only the PMD stimulation caused successful context-specific escape of the kind that we're researching. So it seems at this point, quite obvious that this region is controlling this type of escape. And we then sort of, we hypothesized that the ensemble activity of the cells in the PMD would strongly encode escape as the defensive behavior. So to see exactly what was encoded by the neurons as the mouse behaved, we performed some Miniscope experiments. We implanted a Miniscope and accessed the PMD via this deep implanted grin lens. And the first analysis that we did was this unsupervised classification method. So we just fed a hidden Markov model. This also worked for k-means. We fed the hidden Markov model just the neural data. It had no knowledge of the behavior of the mouse, and had it divide the data into 2 states. So it was restricted to 2 states. And you can see here in this example data that the state for these sample sessions kind of ping pong between 1 and 2. And escape always occurred. Escape is marked by these vertical red lines. Escape always occurred in state 2 for rat and heated plate and CO2, indicating that the neurons in this region as an ensemble were strongly encoding escape behavior. This is what we found across all mice, that you could predict escape at a level significantly greater than chance for all sessions and all assays. So now I mentioned before that there are some downstream target regions of PMd-cck neurons. And we wanted to know sort of what their contributions might be towards controlling escape. That's the AMV and the PAG I mentioned earlier. So to get at this question, we injected artrhodopsin into the PMD and then inhibited the downstream axons at the PAG or the AMV, and for the 3 assays that we're testing. Now you would hypothesize that the PAG would be sort of controlling all types of escape. It's just so well known that it's it's active during all the escapes that we've recorded, and it's known for its role in initiating escape. So we expected this would, in fact, impair all escapes across assays, context-specific and simple stereotypes. And that's indeed what we saw across all 3 assays. Inhibiting the downstream terminals at the PAG decreased the number of escapes. However, we only managed to decrease the number of escapes for AMV inhibition for the two context-specific assays. And this is also something that we hypothesized, given that the AMV is known for its role in spatial navigation. It seemed to have no effect on the number of jumps in CO2, where the knowledge of the environment is not required. So this was interesting that inhibiting the PAG sort of reduced the number of escapes in all assays, while the AMV seemed to be particular to the context-specific escapes. So along with the connectivity this is suggesting between regions, we wanted to see if perhaps the PMD and the PAG and the AMV were, say, more or less synchronized during each of these assays. So if they're communicating more, you would imagine there would be a greater correlation between their signal. So to answer this question, we expressed GCAMP6S in PMd-cck neurons and in all synapsin expressing neurons in the PAG or the AMV. And so this is some example data from the PAG PMD sessions. The PAG is in red and the PMD is in blue. You can see they're quite uncorrelated for exposure to toy rat, and they grow in correlation, increase in correlation for rat exposure. The escape onset is marked by these green triangles. So you can see it's aligned with escape, and the signals are more correlated during exposure to threat. And this is actually what we saw for all of the threat assays for the PAG and the PMD synchrony. They show an increase in correlation for all assays for threat above control. Now, looking at the PMD and AMV signal, you can see a similar thing for the toy rat and rat exposure. The PMD is in blue and the AMV is in orange here. The signals are rather uncorrelated for toy rat. They increase in correlation for rat, shows sort of matched transients just preceding escape here. And we see an increase in synchrony or in correlation from control to threat for the two context-specific assays. But we actually see a decrease for the CO2 assay, the simple stereotyped escape assay. And again, this is sort of what we would expect, as this downstream region seems to be involved in spatial navigation, and no knowledge of the environment is necessary for these escape jumps in CO2. So I'm going to move on to the second paper now, just to make sure we make it in 20 minutes. This was published in eLife also last year. And the motivation for this paper, it had been shown that the PAG controls escape vigor as well as escape behavior in general, as measured by escape velocity. So you can see here in this study by Deng et al. That if you increase the frequency and the intensity of the laser in a channel rhodopsin stimulation experiment, you'll increase the velocity of the escape. So the inputs that may be controlling escape vigor to the PAG haven't been identified thus far. So another thing that we were interested in looking at was that the PMD has been shown to be activated in contexts that are fear conditioned with shocks and social defeat, indicating that it could play a role in coordinating defensive behaviors for both innate threats and conditioned threats. So we studied innate threats in the previous work, and now we were going to look at conditioned threats to see if it was also controlling those types of escape. So the questions we wanted to ask were, do PMd-cck cells control escape velocity? Is PMD activity necessary for defensive behaviors elicited by both innate and conditioned threats? How do PMd-cck cells represent these threats and defensive behaviors during threat exposure? And here are the assays that we use to get at these questions. So the top assay here, the rat exposure assay, is sort of a modified version of the rat assay from the previous paper. But instead of there being a burrow where the mouse could hide, there's a sort of a large empty space here where the mouse can perform various defensive behaviors. And you can see here that this is a sample video. It has more room to approach the rat. It performs these stretch attend postures where it stretches towards the rat to examine the threat. It has more room to escape and express escape vigor, sort of it can achieve higher velocities and freezes at the far wall. So we get a lot of behavior from this. And we see fairly similar behavior for the fear retrieval assay. There's a sort of an approach, stretch, escape, freeze sort of pattern. And we get a lot of behavior to analyze. So how does this PMd-cck ensemble activity encode these various defensive behaviors? We can actually look at a number of behaviors now and compare. We did a similar Miniscope experiment. We expressed GCAMP6S in the PMd-cck neurons and recorded during each of these assays. And interestingly, the only defensive behavior that we could predict above chance level using a 5-fold logistic regression model was escape behavior. So again, the PMD seems to be strongly encoding escape behavior, and interestingly, sort of not encoding these other defensive behaviors that we're seeing in these assays. Moreover, escape could be predicted up to 6 seconds prior to escape onset, indicating that more than just controlling escape or sort of being like an up motor sort of impetus towards escape, the PMD seems to be involved in planning or sort of orchestrating this escape. It's online well before escape begins. So this decoding result, the fact that approach was not very decodable and escape was highly decodable, indicated that the speed towards the threat would probably be not very evident in the ensemble data, but the speed away from the threat would be something that we could decode. So to test this, we use a generalized linear model. This is the testing data here. So we trained the GLM in the toy rat and rat assay, pre-shock and fear retrieval. And we used only speed towards the threat. So only the moments where the mouse was moving towards the threat. And the blue line is the predicted speed and the gray line is the actual speed. And it did a terrible job. We couldn't really predict speed of movement towards the threat. But as hypothesized, looking only at epochs where the mouse was moving away from the threat, we could fairly accurately predict speed for the threat assays specifically, not the control assays. And this was significantly better than chance. So the prediction error of velocity away from threat was significantly below chance for rat and fear retrieval. So altogether, the recordings of this PMd-cck ensemble activity reveal that the cells are highly active during escape. Their activity can be used to decode escape, but not approach velocity. Neural activity could only decode escape, but not the other behaviors. And we thus hypothesized that inhibiting these PMd-cck cells would decrease escape velocity and not affect the other defensive behaviors. So we performed a dread experiment. We expressed inhibitory dreads in the PMD, CCK neurons, and we performed 2 toy rat sessions, pre-shock, and fear retrieval sessions. So here we're showing the CNO minus saline for each of these measures. And you can see that indeed we did manage to decrease escape speed for this manipulation, while nothing else seemed to change. We checked a number of behavioral metrics, like numbers of escapes, numbers of approaches, approach velocity, things like this. And escape speed was the only thing that was sort of consistently decreased or significantly changed between CNO and saline sessions. So next we sought to identify which of the main PMD outputs might control escape speed. To look at this, we did another arch-rhodopsin experiment where we injected the PMD with arch and then inhibited downstream at either the PAG or the AMV. And you can see here that we found similar results for the PMD to DLPAG sessions. We saw a decrease in escape speed, trending for rat, but significant for fear retrieval. And we saw no effect of PMD to PMD to AMV inhibition. And so this indicates that PMd-cck neurons are actually controlling escape vigor via their inputs to the DLPAG, which isn't totally surprising, but it's good to know. So finally, to see if there's a functionally similar pathway that might exist in humans, we looked at the functional connectivity, which can be thought of as the covariation of the bold signal of the hypothalamus and the PAG as participants received aversive stimulation. And the stimulation we're talking about here is just viewing aversive images in an MRI, fMRI. So this data is from the Torwager's lab. There were 48 participants and they were all, they viewed images that were aversive to various intensities. So his lab performed this analysis. They used a predictive model trained on 47 of the participants such that the correlation of the hypothalamus and the PAG were maximized. And then they tested on the remaining held out participant and cycled through all of them. And so you can see here that defining the pathway this way, the pathway showed significantly greater expression for the negative images than the positive images. And interestingly, as the stimulus intensity increased, or as the aversiveness of the images increased, the pathway expression monotonically increased as well. So it seems like there is communication between these two regions that sort of mimics what we saw in the mouse data. It's always exciting when something seems like it might be translatable to the human realm. So to summarize, PMd-cck cell activity is necessary and sufficient for both context-specific and stereotyped escape. PMd-cck cells recruit the AMV specifically for tasks that require context-specific escape, and the PAG for both types of escape. PMd-cck cells encoded and predict escape from both innate and conditioned threats. Inhibiting the cells or their projection to the DLPAG decreased escape speed from a live predator and a conditioned threat. And there's a similar pathway in humans as suggested by fMRI results. So thank you for listening. Thanks to all of my funding sources. Thanks to my PIs, Avi and Jonathan, with mustaches, just an idea. Also thanks to this guy with the mustache, Keanu Reeves. So he was in Speed earlier. I just sort of wanted to throw him in there because he looks really good in a mustache, but he looks good in anything. And then Adhikari Lab, everybody there, the Cow Lab. I appreciate all the conversations we've had and all the help that you've given me. Nathia Suthana's lab, Felix and Jenny, Tad Blair, Garrett Blair, and my committee as well, and my family. So thanks to everybody that has helped me get here, and thanks for listening and having me at this wonderful award ceremony. So that was a whirlwind presentation in 20 minutes. If anybody has any questions, we have a few minutes left, and then we can always do questions at lunch as well. Yes.
I had a question about your Miniscope recording, like the first one that you showed you.
Sure. Yeah.
In the two states, can you tell if it was more defined by different sets of neurons versus the same neurons and their activity?
We didn't manage to find like two sets of activated neurons, and I think it didn't seem like it divided into two groups that were sort of activated for different states. So there was, I guess we didn't really characterize what the difference between states was. We just fed it into the model and determined. But there were definitely cells that were positively activated by escape onset. There were some that were negatively activated. I didn't show you the single cell data because I didn't have time, but I don't believe that there were, like, I don't know, very separate groups that were controlling one state versus the other. We also couldn't co-register across sessions very easily with this data for some reason, so we couldn't see if those ensembles would maintain their identity, if, say, like an escape positive cell maintained that for another assay, unfortunately, but maybe we can do that in the future. Cool. Thank you very much. Oh, come on. Oh, yeah.
Okay. They have to, yeah, they'll do, I don't know, like I said, specifically for your group, and maybe they have it as a separate one for you guys, but generally speaking, that time of year, we've got many people on campus, we'll usually have one set up here and then one as well. And the way we generally do it is that it's assigned seating indoors and guaranteed you're seating inside, and then for comfort. Yeah, I mean, I can always check and see, like, I mean,
are you ready for me to introduce Anna now? Can y'all hear me? Great. I really apologize that I'm not able to be there in person to introduce Anna properly. I'm at a decision-making conference that Anna is going to join me at later today to present her data again. In thinking about how to introduce Anna to you, I thought the one thing that you might want to know about her is that she has been incredibly fearless in her graduate study. So when she joined the lab, she made fairly substantial shift in both methods and in topic. And on top of that, took on a very ambitious set of experiments that led her to some really important discoveries that she's going to tell you about today. And when her questions led to a place where we didn't actually have the methods in the lab to answer those questions, she just set them up and used that to make really important discoveries. And Anna's also been really smart in the decisions that she's made through her scientific journey in the lab. So she's identified some really critical gaps in knowledge, and she's used her fearlessness to just fill those gaps and provide some really important discoveries for a field that she'll tell you about today. So.
Awesome. Thanks so much, Kate. Really appreciate it. Okay, so well, thanks for having me here today. My apologies in advance if some of you might have heard already that I'm about to present to you, but let's dive into it. So here we are. The focus of my graduate work so far in grad school has really been to investigate the basolateral amygdala circuits that act as neural substrates for encoding outcome-specific reward memories. So every day we use stimuli within our environment to tell us about the availability of specific prospective rewards. We also have learned through previous experiences that to gain access to these rewards requires distinct sets of actions. However, in circumstances where rewards or outcomes are not readily observable, we often rely on these previously encoded stimulus outcome representations to enable cues within the environment to elicit detailed representations of specific rewards that they predict. And then we can subsequently use this information to selectively guide our actions and inform our decision-making. And we in the lab can model this sort of cue-directed decision-making process through a task called outcome-specific Pavlovian to instrumental transfer. And so here we train, I train rats that performing two distinct sets of actions gain some access to two distinct food rewards. In subsequent Pavlovian conditioning sessions, animals then have the opportunity to learn that two distinct auditory stimuli are predictive of those same food rewards that they had received during instrumental conditioning. And then in an unrewarded test of Pavlovian to instrumental transfer, animals have free access to both levers, and action selection is monitored in the presence of these auditory stimuli. And because at no point in training have animals ever experienced the presence of these levers concurrently with the presence of these auditory stimuli, this task really requires the animal to, upon cue presentation, retrieve a detailed representation of the specific outcome that that cue predicts, and to use this information to enhance or motivate choice for the action that had previously led to the same outcome. So if animals have successfully encoded these rich, detailed stimulus outcome associations, then upon presentation of these auditory stimuli, they will selectively invigorate their pressing for the lever that's associated with the same outcome as that auditory stimulus, relative to the lever that had earned the alternative or different outcome. And this is called the Pavlovian to instrumental transfer effect. And a focus of my research, and something that our lab is really interested in, are investigating the neural circuits that are particularly important for encoding these detailed associative relationships. So how we learn about cues within the environment and the particular rewards that they predict. And through our research, we found that the basolateral amygdala is really a central hub within the circuitry that mediates this stimulus outcome encoding. Using bulk calcium fiber photometry, I've recorded from BLA principal neurons during Pavlovian conditioning as animals acquire these detailed associations. And what I found is that BLA principal neurons are activated at the time of cue onset, but are also really robustly activated at the time of reward experience. And this is interesting because this is probably a very critical window where animals are actually linking the relationship between the preceding stimulus with the specific outcome that it represents. In some previously published work, we've also shown that this activity, specifically at the time of reward experience, is necessary to encode these detailed stimulus outcome relationships. Optically inhibiting BLA principal neurons at the time of reward experience during Pavlovian conditioning does not actually impair the acquisition of a Pavlovian response policy. So during Pavlovian conditioning, as cues come on, animals are still checking for the food reward at the magazine at comparable rates to EYFP controls. However, inactivating the BLA principal neurons really disrupts the acquisition of the outcome-specific information within these Pavlovian associations, such that they cannot use this information to adaptively guide their choice towards the action that leads to the same outcome as a predictive stimulus. So that's what you see here. Animals who have had their BLA inhibited during Pavlovian conditioning, when tested in Pavlovian to instrumental transfer, upon cue presentation, don't seem to understand which outcome is most likely to be available, and so cannot adaptively choose.
I'm going over to Caitlin's. While she goes and gets Catherine. I set the water, they just reconnected the system.
So,
and I'm running it through. So for the next, until I get back, please use as little water as possible. So that, because it's going to have a backflow.
Robert, can you please mute yourself?
And I don't want the brown water.
okay. So this suggests to us that basolateral principal neuron activity is really important for mediating the encoding of these outcome-specific reward memories. But we also know that cognitive functions don't happen within a vacuum, and that there are circuits within the brain that are important for facilitating these sort of complex cognitive functions. And so we wanted to further explore what inputs might be supporting the basolateral amygdala in encoding these detailed representations. And one region that we thought might be important is the ventral tegmental area, and specifically dopaminergic neurons within the VTA. We know from some tracing work in our lab, as well as others, that the BLA is innervated by dopaminergic neurons. So this is just a stain for tyrosine hydroxylase, which is often used as a marker for dopaminergic neurons. And you can see that there are axon terminals in the basolateral amygdala. We also were interested in this projection because VTA dopamine has sort of a long history of being important for associative learning, and there is a growing body of evidence to suggest that VTA dopamine is also really important for forming more complex cognitive structures. And within those cognitive structures, we have these detailed stimulus outcome representations. So we wanted to see whether or not VTA dopamine might be contributing to detailed stimulus outcome memories through projections to the basolateral amygdala. And to first answer this, I was interested in understanding what the endogenous pattern of dopamine release is in the BLA during the stimulus outcome learning. And so here I used the high affinity dopamine sensor, GrabDA, and monitored dopamine release during Pavlovian conditioning, and found that similar to BLA principal neuron activity, as the animals acquire these associations, you see this robust dopamine release at the time of reward experience, again, during this window of time when animals are likely sort of initially making the association between these auditory cues and the specific rewards that they predict. And so now we know that the VTA dopamine is being released into the BLA at this sort of critical window of time that's important for stimulus outcome learning. Is this functionally significant? So are VTA dopamine projections to the BLA actually needed to encode these stimulus outcome associations? And so here I again used an optical approach where I transduced dopaminergic neurons in the VTA with the inhibitory opsin ARCH-T, and I implant my optic fiber over the basolateral amygdala to selectively suppress those dopaminergic inputs. Animals are trained on instrumental conditioning as previously described, manipulation free. And then during Pavlovian conditioning, I inhibit dopamine inputs to the basolateral amygdala at the time of reward delivery, again, during that period of time where we see robust dopamine release. And recapitulating the findings that we see from inhibiting the basolateral amygdala directly, this doesn't impact the animal's acquisition of a Pavlovian food port approach response. They're still checking the magazine for food during the cue throughout training. However, disrupting dopamine release during Pavlovian conditioning selectively disrupts the acquisition of these outcome-specific memories such that they cannot use that information to adaptively guide their choice in Pavlovian to instrumental transfer. So that's what you see here. Suppressing dopamine release during stimulus outcome encoding later on sort of results in this indiscriminate action selection. And so here we kind of add on to show that indeed the dopamine release that we're seeing in the BLA is functionally significant and necessary for the encoding of these detailed memories. So as any kind of circuit experiments usually go, we next wanted to determine whether or not stimulating dopamine projections into the basolateral amygdala is sufficient to promote the encoding of these detailed stimulus outcome associations. We're particularly interested in seeing whether or not this is sufficient to drive this learning in scenarios where perhaps these stimulus outcome memories might not otherwise be formed. So to address this, we used a Pavlovian blocking protocol. And the idea behind a Pavlovian blocking paradigm is that previously learned stimulus outcome associations can actually block the acquisition of new stimulus outcome associations when those new stimuli are presented concurrently with previously encoded stimuli. So to provide an example of this, I hope everybody here has been to Taco Bell and that through previous experience, you have learned that when you see this sign, you can come to expect delicious, crunchy golden tacos. Now imagine that Taco Bell started planting pine trees outside of all of its Taco Bell locations next to this Taco Bell sign. Because you've already learned that the presence of this sign is indicative of crunchy tacos, the presence of this pine tree isn't really providing you with any new information. And so when presented alone isn't sufficient to evoke a representation of these Taco Bell tacos. So here, the Taco Bell sign, taco association blocks new encoding about pine tree taco association. And so we wanted to use this concept to essentially ask whether stimulating dopaminergic inputs would be sufficient to drive the encoding of these detailed stimulus outcome representations that are otherwise blocked by previous learning. So here, again, using an optogenetic approach, I put the excitatory opsin, channelrhodopsin into dopaminergic neurons and stimulate dopaminergic release in the basolateral amygdala. Animals are trained on instrumental conditioning as I have described. And then during Pavlovian conditioning, animals in this experiment learned that two distinct visual cues are predictive of these food outcomes. In subsequent compound training sessions, these visual stimuli are now presented concurrently with two novel auditory stimuli followed by the delivery of food reward. And the idea here is that because these animals have already learned that these visual stimuli are predictive of these food outcomes, these auditory stimuli don't really provide the animal with any new information. And so these previously learned stimulus outcome associations block the acquisition of these auditory stimulus outcome associations. And I wanted to ask whether or not stimulating dopamine at the time of reward delivery would essentially unblock learning or drive learning about these auditory associations that might not otherwise occur. And what I see is that stimulating dopamine doesn't really impact the animal's entries into the magazine during training, so they're not going into the food port to check for reward any more or less. However, when tested to see whether or not animals have acquired these auditory stimulus outcome associations by presenting the auditory stimuli alone in Pavlovian to instrumental transfer, what we see is that by stimulating VTA dopamine, we are able to drive the learning about these auditory stimulus outcome associations that are otherwise blocked by these previously learned associations in EYFP controls, suggesting that VTA dopamine in the basolateral amygdala is both necessary and sufficient to drive detailed stimulus outcome learning. But dopamine is really a modulatory neurotransmitter and is also likely to be interacting with excitatory inputs into the basolateral amygdala. So we wanted to sort of further explore the circuit by understanding which of these excitatory inputs might be facilitating the basolateral amygdala in this stimulus outcome encoding. And we found that in this previously published paper from last year, that lateral OFC projections, glutamatergic projections from the lateral OFC are really important in facilitating the basolateral amygdala in this encoding function. Optically suppressing these lateral OFC to BLA inputs during Pavlovian conditioning, again, really impairs the acquisition of these detailed stimulus outcome associations, suggesting that this is indeed a very important input for facilitating this detailed stimulus outcome learning. We also know from some previous work that was published by a former grad student in our lab, Nina Lichtenberg, that basolateral amygdala also sends reciprocal projections back to the lateral orbitofrontal cortex, and that these projections are particularly important for the retrieval stage. So animals need these projections intact in order to retrieve stimulus outcome information to enable adaptive decision making. And so the question kind of arises, well, how are these reciprocal connections communicating with one another to support these detailed stimulus outcome associations? And we know that these associations are really multifaceted. They contain information about the identity of these specific outcomes, the associative relationships between the cues and the rewards, as well as cue identity. And so one potential possibility is that these projections are acting within a serial circuit such that lateral OFC to BLA projections enable the encoding of one component of the associative memory that is subsequently retrieved through activation of BLA to lateral OFC inputs. So these reciprocal projections are encoding and retrieving a shared component of the associative memory. Alternatively, these projections might be sort of processing information in parallel, such that lateral OFC to BLA inputs facilitate the encoding of one component of the associative memory, whereas BLA to lateral OFC inputs might be facilitating the retrieval of a necessary but independent component. And so to distinguish between these possibilities, we used a serial circuit disconnection approach. And so here what we did is, as animals undergo Pavlovian conditioning and are encoding these associative memories, in one hemisphere, we inhibit lateral OFC to BLA inputs to shut down stimulus outcome encoding pathway. And then in the contralateral hemisphere, those inputs are left unmanipulated. So stimulus outcome information should be left to be encoded. And then directly prior to Pavlovian to instrumental transfer, when animals have to retrieve these stimulus outcome associations to enable cues to elicit the detailed representations of these outcomes. In the hemisphere where we've shut down lateral OFC to BLA projections during encoding, we now leave the BLA to lateral OFC projections unmanipulated. And then in the contralateral hemisphere where this lateral OFC to BLA encoding pathway was left sort of intact, we complete the circuit by shutting down BLA to lateral OFC projections directly prior to retrieval. And so here, if these reciprocal connections are acting within a serial circuit, in one hemisphere, we have blocked the encoding of the shared associative component, and so there should not be anything to be retrieved. And in the contralateral hemisphere, although that associative information has been encoded, we blocked the retrieval or use of it through inactivating BLA to lateral OFC projections. And we functionally execute this serial circuit disconnection by combining optogenetic and chemogenetic approaches, where in one hemisphere, we optically inhibit lateral OFC to BLA inputs during Pavlovian conditioning. And this is similar to the rest of the findings that I've shown, where animals still go into the food port with this unilateral inhibition. And then in the contralateral hemisphere, directly prior to Pavlovian to instrumental transfer, when animals have to retrieve the stimulus outcome information to guide adaptive decision-making, we use a chemogenetic approach to inactivate BLA to lateral OFC projections. And what we find is that by disrupting the information transfer between these two regions, we sort of impede the animals from using these stimulus outcome associations to guide adaptive choice. And so this sort of provides evidence to suggest that these really are working within a serial circuit, such that the information that's being encoded through lateral OFC to BLA activity is the same associative component that is being subsequently retrieved through BLA to lateral OFC activation. And so to sort of summarize here, I hope that I've conveyed that these BLA circuits are really important neural substrates for the encoding of these detailed stimulus outcome associations, and that the basolateral amygdala receives both cortical and subcortical inputs to facilitate this learning. And with that, I would really like to thank everybody in my lab who's contributed to this work. Some former lab alumni, our collaborator, Dr. Melissa Sharp, for helping us with the Pavlovian blocking protocol, and of course, my mentor, Kate Wassum, who's been really wonderful, and I wouldn't have been able to be here without her. So thank you so much.